Hello, welcome to the iDEC meeting. I'm Tom Ellis, Professor of Synthetic Biology at Imperial College London. That's my labs behind me. My office is up there. I'm not in my lab or my office today because at the time of your event, I'm traveling with my children. But Yang has asked me to give an opening talk for your meeting and record it for you. And so that's what I'll do now. I'm going to give you a 20 to 30-minute talk on my perspectives on how evolution and directed evolution, what you've been working on all year, is critically important for the next 20 to 30 years of my field, synthetic biology. So let me share my screen and give you a bit of my thoughts about synthetic biology and iDEC. So we've had about 20 years now of synthetic biology. And I'd like to sort of dwell on that a bit. The year right now is 2022. It's coming to an end. And we've looked back now, and people have finally stopped saying that synthetic biology is a new and emerging discipline. Now that we've realized that there are students studying synthetic biology that weren't even born when the first two papers from the field came out in the year 2000. So we had papers in that year that described these toy systems of genetic regulators wired up in non-natural arrangements that mimicked the sort of things that we would see in electrical engineering. Jim Collins's lab at Boston University published a genetic toggle switch in E. coli, whereas Michael Elowitz and Stanislav Gleba at Princeton took similar transcription factor regulators in E. coli, but wired them up in a slightly different way to achieve an oscillating network known as the repressor latex. This is sort of the beginning of the field, although obviously it takes a while to get research papers, so you can imagine that the work doing this and work in other labs in similar vein started in the late 1990s, just after the genome sequences started to appear from the first genome sequencing projects. For me, 2005 is really where synthetic biology got exciting. There was a special issue around about November 2005, focusing on synthetic biology, giving some highlights of the field and discussing the first iGEM competitions that had happened and what their impacts had been. This is a really exciting journal issue. It certainly got me hooked. And the one thing within this issue that I really loved was this big page showing a plate of E. coli with the words "Hello World" written on it. This was the E. coli agar plate photo, where light had been shined using a stencil onto this plate, and where the light had been shined, the cells produced a black pigment in response. And so you could produce a writing such as "Hello World." This is actually a remarkably simple system, but really underlined and made me understand what synthetic biology is in that time in 2005. This notion of putting different modules together, modules such as the responding to light and producing black pigment through the enzyme LAC-Z, things that are relatively straightforward to do, but doing them in ways that people had never done before, and particularly doing it in ways where engineering principles were followed. So this really gave me a great feeling about what synthetic biology was about. This was actually work led by an iGEM team in the first ever iGEM competition. And that was a real hallmark of the first decade of synthetic biology, from 2000 to 2010, where iGEM started around about 2004 with undergraduates at MIT, and then branched by the end of the decade into a major competition with teams from all around the world. That was run by the BioBricks Foundation, who also run, in those days, the major meetings. And a biggest first focus from the field of synthetic biology were things such as biofuels, which at the time was an important area for investment. And so that really brought a lot of money and people into synthetic biology in the late 2000s. If I look back and think about that first decade, I myself was a postdoc in Jim Collins's lab in that decade. The things that came out of that decade that really mattered for the field were the ability to do modular DNA assembly. Golden Gate method and the Gibson method were both invented during this time. And the genetic devices was a real focus for the work, understanding how to build switches, counters, oscillators, and start to use recombinases to do logic and memory within cells, particularly within E. coli. And metabolic engineering was really one of the first application areas which got brought into synthetic biology, with groups like Jay Kiesling's group doing strong work on biofuels and spinning off the first synthetic biology companies, and people starting to think about bulk chemicals, specialty chemicals as well, being the sort of things you could make from engineered bacteria and engineered yeast using synthetic biology principles. I started my research group here at Imperial in 2010, which is what we call the second decade of synthetic biology. And myself and Fankang Meng, a PhD student in my group, were lucky that about 2 years ago, we were asked by Nature Communications to write a short summary of this second decade of synthetic biology as part of the celebrations for 10 years of the journal Nature Communications. If you want to see what we wrote about and what we thought were the real highlights of the field, you can go find this as an open access paper online. There's a lovely timeline drawn up here of different achievements that the field made over the 10 years, drawn up by Fankang. But in summary, I think these are the key points, right? We saw synthetic biology go from these toy genetic systems to really much, much bigger and more successful things, such as synthetic genomes and synthetic chromosomes in eukaryotes, from projects from the J. Craig Venter Institute and from the SC2 consortium led by Jeff Booker. We saw groups such as Chris Voigt's group and Howard Salas do automated design of complex systems, particularly logic gate systems, not just in E. coli, but also in yeast and other organisms, so that you can actually feel like an engineer and program something on computer-aided design and then get told exact DNA sequences to get synthesized to have your cells run tasks. Metabolic engineering became far more ambitious and advanced, not just making very straightforward molecules such as biofuels that we would just burn, but instead making big complex molecules such as opioids and rare antibiotics and anti-cancer drugs of really, really high value and high scarcity. Cells started to be engineered and delivered as therapeutics and diagnostics, taking account of the brilliant abilities of cells to sense things in their environment and act upon them. People started putting synthetic biology gene systems into things like CAR T cells to produce better sensing, and engineer things such as gut bacteria that can sense and respond within your gut if you have a medical illness. We even had the first sort of branching into multicellularity, with teams able to drive cells to do pattern formation and to have programs of differentiation of mammalian cells to achieve different states from an initial pluripotent cell at the beginning, given a specific DNA program. This second decade was really enabled by a lot of technologies, and Fankang and I summarized some of them here in this design, build, test, learn diagram. In particular, I think things such as iGEM allow widening education, and things such as AdGene allowing quicker plasmid sharing, and things such as bioRxiv, which allow much quicker dissemination of published material, really helped accelerate the field. But I think it would be unwise not to mention the things that really generated a lot more progress in the field, particularly CRISPR, being able to go inside cells in vivo and edit those and use things such as cheap gene synthesis and sequencing-based measurements to really quickly work inside cells and to make them do the sort of things you want to do in terms of engineering. So we've had 20 years now, 22 perhaps to be more precise, of synthetic biology. So what will this field, what will engineering biology, directed evolution look like if we were to go forward the same amount of time as to what we've just looked back on? So let's think about what synthetic biology might be like and what the world might be like in 2040. Hopefully it will be a better, greener place with a lot more use of renewables and plant-based products in our life and less damage to the environment. Looking forward to the year 2040, about a year and a half ago, I was asked by a German research center to think about the 10 areas of synthetic biology that I would recommend to students nowadays to try to focus on if they want to make an impact beyond the 20 years we've had in the past and think more about what's needed for the 20 years in the future. So this was a fun task, and I'm going to summarize some of this for you first before narrowing down on the one of those things that I listed in my 10 that's highly relevant for you guys this year. This was a fun task to think about 10 areas. And I have to say, I specifically chose areas that were technical areas, not thinking about legal or social areas which need innovation. And I also chose some that were quite obvious and also left ones out that were really obvious. So I wanted to put in a few that people would be comfortable about, but not just put in the same old ones that people would always put in terms of what they hope synthetic biology could do going forward. Two fantastic undergraduate students at the time, Olivia Gallup and Haya Ming, helped me write this down as a research article after I'd first spoken about it as a research presentation. So if you want to know our reasons and my reasons for these 10 being 10 of interest, you can go to this review paper we wrote in the IET Journal of Engineering Biology. So let's delve into 10 areas for the future then. So firstly, and perhaps quite obvious, is industrialization and automation. Synthetic biology, and particularly the act of linking different bits of DNA together to get better gene expression of things like metabolic pathways, is getting to the point now where people can run these things on robots like Opentrons or lab-site echoes. And so it's becoming an automatable task, and including the data collection and the reading and the understanding of that data, and then choosing the next best optimal design. So improving and optimizing your designs, that can now, in many cases, be an automated process. And that's really going to help us industrialize this current thing we do in synthetic biology, which is to get a microbe and to say, I want this microbe to make this, and I want it to make it really well and do more. And so I think that's an area that's going to be of increased focus. It's already been a huge focus of the field for the last 10 to 15 years. But I want to caveat that I think that should really be done at companies, because they're the people who can do this at scale and really understand the automation and build it into their processes, and then reap the benefits of having automation and data collection. Something a lot harder to do in academia. And labs like our own, where we work on limited timescales, and PhDs and postdocs are only ever with us for about 3 or 4 years. The next one on my list, something that since I wrote the list a couple of years ago for the first time, has really become something a lot more mainstream, is the use of machine learning or AI-based methods for designing DNA. Now, I think machine learning and AI is everywhere now, so it's not just for designing DNA, but I wanted to really highlight in these 10 areas of the future that it really makes sense to use machine learning for DNA design. Now, the reason for that is because DNA is just a programming language. It's nature's programming language, with our genomes being our hard drives containing all of the information for running our cells. And over the 70 to 80 years since we've understood DNA and its structure, we have made great progress in learning how DNA encodes instructions in the cell, but there's still lots to learn. And we still don't know precisely what every base pair is doing. But machine learning is a tool that is commonly used for things such as translation of documents, and is getting very, very good at understanding how to translate a legal document written in German into a document that's a legal document in another language, or even it can take a legal document and work out how to say it in a normal language, right? So machine learning is getting very good at understanding languages from first principles and understanding code and being able to work out how to write and code itself. And so I think it really makes sense that machine learning I think very quickly over the next 10 years will become the way we write our DNA, as opposed to the current process of copy paste of modular parts. The third one in my highlights is synthetic cells from biochemistry. And so you may have heard that there are consortia and collaborations around the world doing build a cell and protocell and cells built from biochemicals and vesicles and synthetic life from chemistry. And this is a really exciting area that's growing, and we're seeing great progress in things such as droplets containing purified ribosomes, running reactions, and even now doing things such as migrating and signaling and splitting apart. And who knows, one day all these endeavors may come together and people may be able to get to that point where we throw a broth of biochemicals together in a vesicle and it starts to replicate and divide and replicate and divide. And we will have created life from chemistry, and it would be quite a remarkable moment. And I think we're getting there, and probably will be there by the year 2040. Here's another 3 areas like we proposed in this review. Biosensing, which has always been a common part of synthetic biology, engineering a cell to sense something and respond. But imagine what we could do if we could really hack into all the sensing done by biology all around us in our everyday lives. Could we have biosensing within our clothes? Could we have a way to recognize the sensing going on remotely in the soil in our fields? There's ways we can link something going on in a cell, the way it's sensing anything around its environment. Can we link that to outputs such as electronic ones or even Wi-Fi outputs where data can come to us? Because imagine if we could, it would be very, very exciting. The ability to be capturing huge amounts of data from what the microbes and from what our own cells are seeing and feeling all around us in the world. That's an area that can only get bigger and more expansive and could really solve a lot of problems for humankind. The next one down on the list is engineering organisms for sustainability goals, because I just mentioned humankind's benefits, but we should also think that about humankind's interactions with the earth and the environment. And I think that every one of you as young students, certainly all the students I'm teaching at Imperial, really want to go into this field of working in engineering biology to give a benefit and to improve the environment, much more so than improving the lifetimes of old rich people through new medical treatments. I think it's important that we start to really focus what we do in synthetic biology on sustainability, recycling of materials, production of fuels and materials without the need for fossil fuels, capture of CO2, sustaining niches in ecology and helping increase biodiversity through any means we can, including GM microbes and GM organism means as well. That's a really big one. I hope we can make progress on that. A couple others to go for: cellular communities and multicellularity. I think we've done a lot of work in synthetic biology just thinking about single organisms and what they can do and how we can program them. But really to do the cool stuff that biology can do, if you think about multicellular organisms, fish, corals, strange fungi, and definitely plants, right? They do it with lots of specialized cells doing different things in parallel to their main core cells doing a variety of essential tasks. I think we really got to get a lot better in engineering biology in terms of using more than one cell and not just having monocultures of engineered cells. Another few areas to list: synthetic genomes, an area my own group work on. I think that is clearly going to be getting bigger, more ambitious. We've gone from 500,000 base pair chromosomes in the smallest bacteria done by the Venter Institute in 2010 to now having the E. coli genome, synthetic with many thousands of genes recoded within it, work by Jason Chin's group. The synthetic yeast genome will be coming very soon. And who knows, by 2040, we could be well on the way to synthetic mammalian genomes or plant genomes through this approach. And so we may be thinking next about customizing genomes or having dynamic genomes that can delete regions and bring in regions at different times to have different specialization. Artificial cells is one that I already covered, and is here at number 8 again as an area where we think we can have cells that you can bring in tasks and make this vesicle that can become synthetic life. So imagine once we achieve that, what are we going to do with that? We will have something that can do what an engineered microbe can do, but maybe without the worries of it being a GM microbe, and maybe with more control of its evolution, maybe with less control of its evolution. But it'll certainly be an exciting area. And the last one on this list is materials with DNA encoded properties. That's an area, again, my group work on this notion of growing materials rather than harvesting them from biology, and perhaps the abilities to write programs in DNA, into plant cells, into bacterial cells that make copious amounts of materials, and have those programs alter the way and the materials are made so that they have different material properties, or perhaps even be living materials that can respond to the environment, regenerate or change. Wouldn't it be great if our iDEC t-shirts one year changed color depending on our mood, or depending on the weather, or depending what award our team won? So the last one, whilst we go to the topic of iDEC, is the one that really matters for you guys, which is the real-time precise control of evolution. So this is one that I really thought about before putting on this list. And I thought, what in synthetic biology what tool, what methodology would we like now that we've got CRISPR, right? Because in 2010, when I founded my group, we were making better. We could see gene synthesis would get cheaper. We could see that Golden Gate Assembly and Gibson Assembly was making DNA assembly better. So we could see that we were going to get there inside cells, but we still had this barrier that once you put stuff in cells, you have to see how it works. And then if it doesn't work very well, you've got to repeat that process. You couldn't edit inside cells. But then CRISPR came along, and we all became people who could edit with inside cells. And that has transformed synthetic biology and a lot of biotech as well. So I had to think, making this list, what would transform the way we do engineering biology in the next 20 years? And for me, nothing could transform it more than the ability to bring evolution into the way we do the engineering, to use it as a tool. Now, this would set us apart in all of the other branches of engineering, which focus on modular parts, computer-aided design, to put well-known parts together in combinations to build something like a car or an airplane with the hope that it's going to work one of the first 2 times. When we do this in biology, we can take that same process, but actually we kind of have the benefit, particularly with microbes, in that there's millions of cells in the tube in any one time. So we can try out many different thousands of designs at one time. And people are doing that and now letting selection start to choose which are the most appropriate designs out of a single tube. The rest of engineering doesn't do that, right? They don't make a million different airplanes, try to fly them all at the same time. And the one that flies the longest gets to be the airplane that flies you to the other side of the world one day. So we can show that we can really do selection from millions at the time. But these cells evolve and mutate, so they do mutation and then selection and evolve. So they have this as a dynamic process. And the process of evolution gives us everything. And it really makes something that's not very good over time be something really special. And I think, anyone who's played Pokemon knows about evolution and can see the benefits of evolution over time. And I think now we're at this really exciting point where we want to try and bring this in as the main tool for the way we do synthetic biology, much more so than computer-aided design and automated design. Now that we have new methodologies like CRISPR and a better grasp of what goes on inside cells and how we can manipulate that, we can really do something exciting in synthetic biology with evolution over the next 10 to 20 years. So just to cover some of the technologies that I find really exciting in this space, the ones I think that really are important that might be allowing us to start doing real-time control evolution inside cells are the ones that allow us to do targeted mutation and to do controlled selection. So for targeted mutation, we have methods such as MAGE, multiplex automated genome engineering, where we can target certain regions to get mutations. And we also have things like OrthoRep, a specific system in yeast where a set of genes can be put on a special piece of DNA that mutates far, far higher frequency than the rest of the yeast genome. Other tools that try to target mutation to specific regions of the genome are out there now. There are mutators where genes such as cytidine deaminases are fused to things such as T7 RNA polymerase. So you can send an enzyme that will mutate DNA to a specific region of the genome and have it processed backwards and forwards through that region, mutating that area far greater. So you're really able to target one gene within a whole genome, or even multiple genes, which might be worth doing. And the Evolver system from John Duber's lab does similar, but with our friends from CRISPR, Cas9, going to targeting specific locations and making those areas hotspots for mutations inside mammalian cells, inside E. coli, and even in yeasts as well. So we have a whole variety of tools for targeting mutation. And there are also lots of tools now appearing that are pretty cool for helping us control selection. One of them with the same name as the last tool we saw, also called Evolver, but with different capital letters and different small letters, from Mo Khalil's lab, a microfluidic, millifluidic system that helps us to have control over cells and the directions of their selection in response to pressures. If you have a lot of money, you can buy a Berkeley light system, which can allow you to do control over individual cells and which ones get selected and which don't, all the way into mammalian cells. And there's also genetic systems that you can start to use for controlling selection. One of which I love here, which is the newly published system RADARS, where CRISPR is used to measure whether an mRNA is present inside a cell and being expressed at a certain level. And if in response to that mRNA being expressed, it can then trigger the expression of any other gene downstream, something like a GFP or an enzyme can be triggered. So that means you can have a process where mutation might lead to more of a target gene, and you can immediately have that being linked to a selection of your choice, such as an antibiotic resistance. And so we're already seeing the first papers where targeted mutation and controlled selection are coming together with automation to give us this sort of next generation of directed evolution. Things such as the PRANCE system from Kevin Esveld's group at MIT, with automated phage display evolution campaigns running over weeks using robotics. And the ACE system, automatic continuous evolution, which brings together that OrthoRep system I told you about in yeast for continuous targeted mutation to certain regions of the cell's DNA, along with the automated selection of the Evolver millifluidic system, allowing the two to come together and us to have control. And so we know from Pokemon that if you can target your mutation and you can control the selection, you can choose your organism and what specialization it can get. You can learn those tricks. So that's really where I think synthetic biology needs to go next, with a lot more use of in vivo directed evolution. So we've had 20 years of synthetic biology. And I think it's actually a bit of a shame that really directed evolution is only being talked about now as a major goal of the field to take it forward after 20 or even 22 years. Because directed evolution itself is over 30 years old as a subject. And it's staggering that you guys doing these competitions, that we haven't had competitions like this before. Phage display from Greg Winter's lab at MRC LMB in Cambridge came out in 1990 and has been a process ever since then, and is highly important in manufacture of antibodies. And the directed evolution that most people think of, indeed probably most of you guys are thinking of it, the sort of stuff Francis Arnold's lab do with PCR, improving enzymes, that's been around since almost exactly 30 years ago. If you look at this first key paper, which I'm showing up here, you can see that the paper was submitted in September 1992. So 30 years and 1 month from now. So let's think then, whilst you're enjoying iDEC, then what you could do 20 years from now, if you want to do the next 20 years of synthetic biology, or even 30 years from now in the year 2050, if you want to think about everything that's been achieved from directed evolution since it was first invented and where it could possibly go in terms of its abilities to change biology and help save humankind and improve our relationship with the environment and Earth in the rest of this century. So thank you all for listening. Enjoy iDEC. And I'm sorry I can't be with you, but hopefully it's a really exciting and rewarding meeting today. Thank you very much.